Hi there. I'm Tim Ogden, president of the Bardet-Biedl Syndrome Foundation. And joining me today is Ben Shaberman, who is vice president of science communications at the Foundation Fighting Blindness. And I want to talk to Ben about research on visual impairment treatments, because the Foundation Fighting Blindness is one of the leading funders of research all the way from basic discovery all the way through clinical trials. And so they're really have their finger on the pulse of what's happening across the board on research, ultimately towards finding a cure for visual impairments, whatever the cause. So thank you for joining me, Ben. I'd like to start just with an overview, because there's a long process from discovery to something that's actually available, approved for treatments. And the news headlines that you see sort of pop up often are about some big breakthrough in mice. Could you walk us through how something goes from a headline on Yahoo News about a big breakthrough in mice to where it gets to potentially available to people as a treatment?
Sure, and I'll comment that it may take several years to get to the big breakthrough in mice as well, because you have to, if you're looking for the root cause of an inherited retinal disease, you have to know the genetic defect. You have to know how that's impacting the retina. You have to create the mouse model. Then you have to create the therapy and test it in a mouse. And that's the point that you're talking about, where there's this big breakthrough. But the process from maybe some initial efficacy in a mouse model to an actual clinical trial, that can take several years. Usually that first experiment in the mouse where there's some efficacy, usually you still have to iterate a few times to get the dose right, make sure you know where in the retina it needs to go. And, with something like a gene therapy, there are different elements of the gene therapy, and you may want to tweak those. So you'll iterate a little more to make sure you get the optimal formulation and administration procedure. And then for the next perhaps couple of years, you'll go through what we call the safety and tox studies, where you want to make sure the treatment is safe and well tolerated. And you'll probably do that in a large animal. And in some cases, the large animal may be an animal with the same retinal disease, or it may just be a large animal that has no retinal disease that you're using for safety and toxicology studies. So that takes some millions of dollars, maybe a few $1,000,000, and it can take a few years. And then hopefully at that point, you have all the documentation you need to file what we call an IND, an investigational new drug application. It's a really lengthy document. It's got your clinical trial design, your manufacturing process, all the animal study results. You submit it to the FDA, and then the FDA hopefully authorizes your trial.
And then, on the trial side, though, there's phase 1, phase 2, phase 3.
Exactly. So the trial side typically takes about 8 to 10 years. It's expensive, usually tens of millions of dollars for a drug addressing a large population. It can be much more than that. But there are essentially 3 phases to a clinical trial, if you want to simplify the discussion. Phase 1 is safety. And phase 1, you just want to make sure the treatment is doing no harm and it's well tolerated. Depending on the treatment, you may do that in healthy subjects, or you may do it, if it's a gene therapy, let's say for a retinal disease, you're not going to put a gene therapy in a healthy eye. So you'll do your phase 1, and actually that'll become a phase 1/2 in a small cohort of patients. And again, your focus is safety. There may or may not be an efficacy signal if you're doing a phase 1/2 and including people that have the disease. And then from there, if the treatment performs safely, Then you'll move into usually a phase 2, where you're really trying to get your initial efficacy signal. It's a bigger group of people. You've learned more about the dosing at that point. You may still experiment with different doses. You've learned more about the administration procedure, especially if you're talking about gene therapy or a cell-based therapy. And that phase 2 cohort will be a larger group of people. You may expand to a second center, a second clinical trial site. If you get some good results there, then you'll move to a phase 3. That's your pivotal phase. That's your largest cohort. That's where the treatment that you're using will be of a clinical grade similar to what you're going to use. Hopefully, if you get FDA approval, you will most likely have multiple sites that will be involved in the trial. You want to make sure that each site can replicate those results that you're looking for. And again, a bigger cohort overall. And then once you're done with the phase 3, if you've met your endpoint, your primary endpoint, then you'll go to the FDA and seek approval. If it's a drug, you'll submit a new drug application. If it's a biologic, like a gene therapy or stem cell therapy, you'll file a BLA, a biologic license application.
So I mentioned the Foundation Fighting Blindness is a major funder of research across the board. Yeah, how does the FFB allocate funding between sort of the basic side, the translation from animal to human, the clinical trials? How do you decide what to invest in?
Right, that's a great question. And we do fund every stage of research. We still fund a lot of basic research. We fund translational research to help move things toward clinical trials. We fund some natural history studies. We fund no-cost genetic testing. But generally speaking, about 25 to 30% of our core budget is going to translational research. We have what we call our Translational Research Acceleration Program. And that's specifically to move things toward that IND that I mentioned earlier. And those projects tend to be a little more expensive than the basic research projects, because as you move into perhaps a larger animal or a more involved research, it does get more expensive. But one thing I'll add, we have another program called our RD fund, our Retinal Degeneration Fund. This is a venture, what we call a venture philanthropy fund, where we're actually investing in companies who are either ready to move into clinical trials or close to moving into a clinical trial. We've invested more than $100 million in somewhere in the neighborhood of a dozen companies. And so a lot of that investment is translational as well, because if the company isn't in the clinic, they're at a translational stage. So one last comment, what's so important about translational research and that early clinical research is attracting outside investment from biotechs and pharmaceuticals. And we're really focused on that because the biotechs and the pharmaceuticals usually like some proof of concept, especially in an early clinical trial before they invest. So that's our focus, to get things to the point where we can start attracting the bigger dollars from the biotech and pharmaceutical industry.
And that's my understanding, Ben. Just want to make sure that my mental concept is right, is that translational side is something of a valley of lack of dollars, where the NIH funds a lot of basic research, but doesn't fund translational research. And the biotechs and the pharmaceuticals, it's so high risk, that translational phase, that they're not willing to invest until you've shown, particularly for a rare disease, that there's some likelihood that this approach is actually going to work in humans and not just in mice or fish or other small animal models. Is that right?
That's exactly right. And, the same holds true for venture capitalists who might be investing in small companies. They really like to see some signal in a clinic beforehand. But with our program, because the foundation, if I can lack a little humility about the foundation, we're recognized as leaders in understanding the space. And when we invest in something, we can often attract investment from venture capital firms because they like the fact that we've put our stamp of approval on it. So a lot of times when we invest in a company, we attract other investment from venture capital firms. That's been a model that's actually worked well for us.
So I mentioned a second ago, that particularly in rare disease, and there are a lot of causes of visual impairment, and Foundation Fighting Blindness is fighting, it's fighting blindness, not specific causes of blindness. Can you tell us a little bit about the most common causes of blindness and where they fit, and the more common inherited retinal degeneration diseases? What are the numbers, the percentages between these various causes?
If you look at blindness globally, the major causes are things like age-related macular degeneration, which is, of course, not rare. And we as a foundation do fund dry AMD research. Glaucoma is a big cause. Some forms of glaucoma are treatable, others are more difficult to treat. In underdeveloped countries where medical services are not readily available, cataract is a huge cause as well. Obviously, in this country, most people can get cataract surgery. It's relatively safe and it's outpatient. It's pretty simple. In really poor countries, vitamin A deficiency can be a cause, but those are the leading causes. AMD, diabetic retinopathy, glaucoma, cataracts. As far as the inherited retinal diseases are concerned, if you look at the US population, there are about 200,000 people affected with a rare inherited retinal disease. That extrapolates to more than 4 million people worldwide. But in the US, you take that 200,000, and about 100,000 of those people have RP, retinitis pigmentosa. About 30,000 have Stargardt disease. That's the inherited form of macular degeneration. Another 25,000 have Usher syndrome. That's RP with hearing loss. And then that additional 40 or 50,000 is split up among a lot of less common diseases: Leber congenital amaurosis, Bardet-Biedl syndrome. The list goes on: retinoschisis, choroideremia.
And so, for context, for people from the Bardet-Biedl syndrome community, just to think about that, the estimate is there should be somewhere between 3,000 and 5,000 people affected by Bardet-Biedl in the US and Canada. So in relation to that 200,000 that Ben is talking about, we think there are about 3,000 to 5,000 people in that 200,000 that are BBS, compared to, say, 100,000 for retinitis pigmentosa. But Ben, I imagine there are some people listening who are a bit confused now, because historically, a lot of people with Bardet-Biedl syndrome were told that their retinal degeneration was retinitis pigmentosa, that retinitis pigmentosa is a symptom of BBS. And I understand that confusion because the phrase, the words mean slightly different things and are used in multiple ways. Could you walk us through a little bit about what retinitis pigmentosa is in terms of a type of retinal degeneration, and what it is in terms of a genetic cause of retinal degeneration?
Sure. So retinitis pigmentosa, I think for people to understand, is a label, really. And there are some ambiguities and contradictions in that label. I'll say that right off the bat. But generally speaking, if you think about the retina, the outer retina is where you have rods and you get your peripheral and night vision. The center of the retina is the macula. That's where you have your cone vision and visual acuity, daylight vision, etc. And generally, RP is when the outer retina is affected first, and people have night vision and peripheral visions, and vision constricts over time. And then eventually, in many cases, central vision is affected. So I think that's a really good generalization of RP. And I know with BBS, that's what can happen. And there's actually some overlap in the genetics. There are some genes that cause Bardet-Biedl syndrome when they're mutated. Those same genes, if they're mutated in a different way, can also cause non-syndromic retinitis pigmentosa. Now, I can tell I'm probably starting to confuse people as well, but the way a gene is mutated is really important. But without getting into too much detail, the way RP affects the retina can vary widely, but there are definitely some similarities between the way BBS affects the retina and RP affects the retina. And I think it is fair to say, generally speaking, that the two are very closely related in how they affect the retina and cause vision loss. The one caveat is that there are people who quote unquote have RP, but have more cone involvement early on. They have more central vision loss. And my understanding is that same thing happens with some Bardet-Biedl syndrome patients. So these labels are really an imperfect way to characterize these diseases. But yes, RP and BBS are very similar.
So given that, when we see things about genetic therapies or gene therapies for RP, do they apply potentially to people with BBS, or is that a completely separate population, that 100,000 that we talked about, and the gene therapies for RP? How should people with BBS think about that work?
Right. And so when we talk about a gene therapy, we're often talking about a gene replacement therapy. So when you're talking about a gene replacement therapy, you're identifying the mutated gene in the patient. And I'm just going to give an example. Let's say it's rhodopsin in an RP patient, RHO. Well, then what you need to do is replace that rhodopsin gene, that mutated version. It can get more complicated than that. And rhodopsin is a choice that could get more complicated. But anyway, that's gene replacement. Now, there are like 80 genes that can be associated with RP, and I know it's somewhere in the 20s for BBS. So to have gene replacement for all these different forms of RP and Bardet-Biedl syndrome is a pretty big challenge. But there are other forms of gene therapy and other therapies in general that we call gene agnostic that are designed to work independent of the gene causing the disease. And in those cases, a gene agnostic therapy, maybe for RP patients, might also help Bardet-Biedl syndrome patients as well.
Let's talk more about that gene agnostic therapies, because as you say, there are more than, I think, 26 different genes currently identified for BBS. And we know that there are about 30% of the BBS population that we still don't even know what the gene is. But those are going to be 10, 12 patients total in the world, right? A gene-specific therapy for those people is probably pretty unlikely, just as you were talking about the millions of dollars it takes to go through each of those steps. So, gene agnostic therapies are probably a lot more relevant, particularly outside BBS 1 and 10, which account for about half of the BBS population. So could you tell us more about gene agnostic therapies? What are those? What are they doing? Where are they in process right now?
Right. So a really exciting example of a gene agnostic therapy, and it's just moving into a clinical trial as we speak, is it's a gene therapy, but it is gene agnostic, and I'll explain how it works, from Sparing Vision. This is a French company. So earlier we talked about RP. The typical course of disease is rods are affected first, the outer part of the retina. And over time, as rods degenerate, as people lose more and more rods, then they're at risk of losing their central vision and their cones. And what researchers in France observed is that rods they produce a protein that helps keep cones alive. And they discovered this protein about 20 years ago. And they very aptly call it rod-derived cone viability factor. And what they've done is they've created a gene therapy, launched the company Sparing Vision, that expresses this rod-derived cone viability factor, which the acronym is RDCVF. So the idea is you're trying to save cone vision in people with RP. And the initial clinical trial, which will be taking place in Pittsburgh and Paris, that's where Sparing Vision has a lot of activity, will be for RP patients. But over time, maybe once, if this gets FDA approved, fingers crossed, knock on wood, they could expand it for other people who have these conditions where loss of rods puts people at risk of loss of cones. And again, independent of the mutated gene. Now, there's another approach already in a clinical trial, actually 2 clinical trials, to address oxidative stress. So in all inherited retinal diseases and age-related macular degeneration, oxidative stress plays a big role in the retinal degeneration and the vision loss. And so researchers at Hopkins looked at an already approved, FDA-approved molecule called N-acetylcysteine. We'll call it NAC for short. They looked at it in animal models. It slowed degeneration. It basically stopped the progression of disease. They did a little clinical trial in humans, and they're launching a phase 3 version of that trial for RP patients sometime this year. We at the foundation invested in a company that's modified that molecule, NAC. They've added an amide to it. It's called NACA. That's actually in a clinical trial in Australia for people with Usher syndrome. And we're hoping to get a readout on that in a couple of months, hopefully September, October. If that looks promising, they'll open a site in the US, again, for RP patients. But it's gene agnostic. It's designed to work regardless of the mutated gene. So those are 2 really good examples. A third, really quickly, is optogenetics. That's bestowing light sensitivity to a retina that's basically lost all or most of its ability to process light. Again, gene agnostic. It's a big focus for us as a foundation because of what we've already talked about. There are, when you take all the inherited retinal diseases, it's near 300 genes, when mutated, that can cause IRDs.
One follow-up question there on optogenetics, Ben, because that's always confused me. Why it's optogenetics? Because it does, like, it's gene agnostic. Why is the word genetics in
Right. All that means is you're using a gene and ultimately a gene therapy to express the therapeutic protein. So in optogenetics, what you're doing is you're enabling cells in the retina to express or to process light, to become light sensitive, cells that normally aren't light sensitive. So the gene isn't replacing a bad gene. All it's doing is it's empowering the cells to be light sensitive. That's what optogenetics means. It means empowering a cell, usually a neural cell, to respond to light. And optogenetics is actually being used in the brain for things like depression and Parkinson's disease and other things. They're using light to activate certain things in the brain. I don't know much beyond that.
Yeah.
Once you get into the skull, I'm kind of lost. So,
Ben, if you'll permit me to editorialize for a minute here, because I think there is a specific question for BBS families, and please don't hesitate to tell me I'm wrong along the way. But for some of these gene agnostic treatments, I think a lot of people in the BBS community would say, well, I want to get into those clinical trials that for gene agnostic. If it's gene agnostic, it shouldn't matter. But certainly my perspective on this is that the companies developing these are very wary of syndromic diseases in gene therapy trials, because it's just another variable. They're so focused on showing safety and efficacy in the simplest way possible to get through that process as quickly and as cheaply as possible. Right. And the people with syndromic diseases introduces another, like, other things that could go wrong that then slows down the whole process. Is that largely right?
That's very true. And what I'll encourage people to do to understand this better, if they go to clinicaltrials.gov, they can look up some of the trials for retinitis pigmentosa. And there's what they call inclusion and exclusion criteria. And you'll see in the exclusion criteria things like glaucoma, cataracts, but there could be other conditions, diabetes, whatever, for exactly what you're saying. They want to limit the variables. But the hope is, and the expectation is, once you get something through a clinical trial, then these companies are going to be motivated to get this to as many people as possible,
right? Because
they want to sell the therapy. So we hope and we expect that the range of conditions will be expanded. But yeah, in clinical trials, they want to pick the people that are most likely to respond and least likely to have an adverse event or safety reaction. And honestly, if clinical trials are trials, it's research. So in a way, if somebody has a lot of symptoms and a lot of issues, maybe it's kind of good they don't go into an early clinical trial and expose themselves to risk. Now, a gene therapy, that's just contained in the eye. For like NACA or NAC, which we talked about, that's a systemic drug. And people may want to be careful if they have gastrointestinal issues, liver issues, whatever. So it's, ultimately what the FDA wants to do in these trials is protect the patient. That's first and foremost. So as much as people want to be in trials, there are some good reasons not to be for safety.
And that's an excellent point that, we all, for our children, for our loved ones, want this to go as fast as possible. And you typically don't hear about the trials that fail. The trials that get shut down don't get reported in the headlines unless they're truly disastrous, right? The ones that just don't work all that well, or there were some adverse effects, they don't make the front page of the news. It's only the ones that work. And so important for people to remember, as you said, these are trials. There are risks that go along with these trials. It's not just how quickly can I get in to get the benefit involved. Ben, what's the best way for people to keep up with the Foundation Fighting Blindness and the research that you're funding and that you're tracking?
Sure. So definitely our website, fightingblindness.org. Part of my role, in addition to doing webinars and presentations and so on, is to write articles on emerging therapies. And those are all posted to the Foundation's website. I host a podcast called Eye on the Cure, and that you can get to from the webpage as well. That, depending on the guest, can be pretty informative as well. Like a couple episodes ago, I interviewed Jamie Kern from Nacuity, who is they're developing NACA. And so we talked a lot about that drug. You can follow us on Facebook. We have a lot of webinars and presentations. And we have a pretty robust chapter network throughout the US, somewhere in the neighborhood of 40 local chapters. And we do local events there. We have speaker series, and that's where people can meet other people that are affected by inherited retinal diseases. As I'm sure many of the members of your community can relate, they probably don't know other people affected just in their general circle of people by an inherited retinal disease. But you get involved in the community, like your foundation, then you meet other people. And that can be both emotionally a great thing and logistically and practically speaking a great thing, because you learn from other people. So, but that's a great way to stay up on education as well.
And, just reinforcing what Ben said, that's why, for most of the people watching this video, it's because they're attending a community event to connect with other people with BBS. But I can also endorse what Ben is saying, is it's not just people with BBS, that a lot of these challenges of visual impairment are shared across a variety of causes of visual impairment. And there's a lot to learn from the overall community about resources available, about coping with vision loss, about accessing services and those sorts of things. So I'll say, personally, I found the original BBS Family Association through an FFB conference where I met the first other family, back in the olden times when the internet was young and it was really hard to find people. So, and finally, I'll note, Ben is also the author of a book called Retina Boy, that is a science fiction adventure about a boy who's born without retinas. And so if any of you are looking for some fun summer reading material, I'd encourage you to check out Retina Boy. Ben, thank you so much for your time and helping walk us through some of these very complicated things that we want to keep track of.
It's my pleasure. I'm honored to be connecting with your community. And just a last comment, the more patients and families can advocate, the better. So, make your voices heard, be out there, educate your friends, family, connect with the foundation to let people within the foundation know more about BBS. There are a lot of these inherited retinal conditions. So it's helpful to advocate within the IRD community as well. So I applaud you for what you're doing, and Tim for leading the BBS Foundation to organize families and advocate and move forward to some solutions.
Thanks, Ben. Thanks everyone for watching. We'll look forward to talking to you soon.
Thanks.